forensic science has evolved into a field that incorporates expertise in other fields such as chemistry, anthropology, and entomology, as well as the scientific method to investigate and solve crimes. However, much like any scientific profession, we can find errors in forensic science which may lead to erroneous conclusions and testimonies. This can ultimately lead to the incrimination of an innocent individual. In this video, we will educate our viewers on the errors within different disciplines of forensics and its potential effects through the examples of real-life forensic cases. Two types of errors that are prevalent in any scientific profession, hindering accurate results, are human error and technical error. Negligence and incompetence comes from a lack of training and level of expertise. Bias is the cognitive process that affects how one makes decisions based on their personal beliefs and opinions, often not based on logic. For example, a forensic expert may be inclined to believe that two fingerprints match because they heard from police that the suspect is guilty of the crime. Technical or instrument error is the difference between a measured value indicated by an instrument and the actual value. This can be seen in analytical instruments with an acceptable level of error, like a balance scale, or in instruments with limitations such as insensitivity or detection. Errors specific to forensics are misinterpretation and false testimony. There are methods in forensics that have limitations because they're based on subjective interpretation, such as matching of similar bullets based on striations, or fingerprints based on patterns. There are no scientific standards that define a pattern between two pieces of evidence as a match, so scientists must base their conclusions on observation and experience, which may lead to erroneous interpretations. Finally, forensic testimonies can be exaggerating by suggesting evidence is rare or unique, implying that the suspect is the source of the evidence. In the same regard, it can be downplayed by saying that an analysis is inconclusive, when in fact it should exclude the suspect.
These are the most frequent forensic methods involved with wrongful convictions, but we will focus on serology and hair analysis. For the first case, we are going to talk about Timothy Cole, who died in a Texas prison in 1999 while serving a 25-year sentence for a rape he didn't commit. Nearly a decade later, DNA evidence from the posthumously exonerated Cole and implicated another man as the perpetrator. Michelle Mallon, the victim, testified for the state and identified Timothy Cole in court as the perpetrator. A forensic examiner from the Texas Department of Public Safety testified an analyst in his lab had examined the rape kit and collected from the victim at the hospital after the attack. He said that the test had determined that sperm was present on the swabs, and the analyst told the jury that both Mallon and Cole had type A blood. Mallon is a secretor, as said by the forensic examiner, meaning that her blood type can be determined from bodily fluids other than blood. The analyst also testified that his lab had compared foreign pubic hairs collected from the victim's underwear and body during her hospital examination. He testified that the hairs had similar characteristics to Cole's pubic hair, but said the analyst conducting the test could not make a firm conclusion because there is no adequate empirical data on the frequency of various class characteristics in human hair. The analyst's assertion that hairs are similar is inherently prejudicial and lacks probative value.
Another case that deals with sources of error is seen in the Dewey and Gerald Davis case, where forensic analyst Fred Zane gave a misleading testimony and misinterpreted the results. Gerald Davis and his father Dewey Davis were convicted in 1986 of kidnapping and of sexual assault. Fred Zane had tested the semen for an ABO blood type which had characteristics of a person with type A blood. However, he suggested that the blood type was from contamination and that the contributor was actually type O, which happened to be the blood type of Davis. Today, this method is no longer used and has not been used for more than 35 years. Gerald and Dewey were both found guilty and spent 8 years incarcerated, but were later exonerated through DNA profiling. For the Cole and Dewey Davis case, we witnessed the unfortunate reality that errors do occur, leading to the wrongful incrimination of innocent individuals. Errors are not exclusive to forensic disciplines, but can also arise in other scientific professions. These errors primarily come from human and technical factors, which include negligence, bias, incompetence, and instrument errors. Within the field of forensics, misinterpretation, eyewitness misidentification, and misleading testimonies are the most prevalent. These errors can significantly impact the accuracy and reliability of forensic findings and testimonies. However, it is important to recognize and understand these errors in order to address the underlying problems and prevent the wrongful conviction of innocent individuals.